Looks like there was a Zoom outage this morning. I sent an email, but it looks like it just sent. So my apologies for not checking. Um, yeah, so we'll go ahead and do the lecture right now since we were unable to do the lecture this morning. And I'll go ahead and go right into lab material as well. Okay, so it's pretty much the same content. The labs have already been assigned, but I'll cover the material right now as we go through the lecture. Okay. So yeah, thank you for being patient. And I do apologize about this morning. I haven't heard of limbs going out yet, but yeah, it's new to me. Okay. All right, so we'll get started. So chapter 3, molecules of life. This chapter is quite long. This has a lot of the base molecules, or a lot of the core ideas that are going to be talked about throughout the rest of the semester. Okay, so yeah, molecules of life include sugars, fats, lipids, protein, DNA, RNA. Those are the main molecules that we're going to be talking about, right? Covalent bonding here, right? We talked about the different types of chemistry. Today we're really talking about covalent bonding as well as hydrogen bonding, okay? All right, so why macromolecules? So biological systems utilize a few very distinct types of molecules. These molecules are mostly made of carbon, nitrogen, oxygen, and hydrogen, right? We've seen that the majority, the percentage of your body weight is made up of these elements here, okay? These molecules are considered organic compounds, okay? Organic compounds are typically referring to compounds that have a base structure of carbon, okay? So we call it carbon-based life or organic compounds, right? And remember, carbon is special because it's an element that has the ability to form 4 bonds. So it makes it a very unique and important element in that it can form those 4 bonds, and it can make a very good structural element for these biological molecules. Okay, I like to think of it as that like fits all Lego piece that kind of ties everything together, and you'll see that when we look at the different molecules and where these carbons are located and how they're important. All right. All right, so carbon chemistry, variations in carbon skeletons. Notice that each carbon atom forms 4 bonds and each hydrogen atom forms 1 bond. One line represents a single bond sharing the pair of electrons, and the 2 lines represents a double bond. So you'll see double bonds here in the hydrocarbon skeletons, right? So here we have double bonds. So this is a benzene ring, a base of a lot of different molecules or biological molecules, okay? So you have branched or unbranched hydrocarbons, right? And these hydrocarbon skeletons are going to be the main base of a lot of different organic compounds, right? And typically what we see for these compounds is that many of these compounds are not polar, right? So remember, in order to mix with water, you need to be polar, right? You need to have a charge. These molecules right here really don't have a charge, okay? They don't have anything that gives them a negative or positive charge. Therefore, they're not able to mix in water, okay? And that's important to note, right? Hydrocarbons, saturated hydrocarbons. What I mean by saturated is that the carbon is completely covered with hydrogens. All the bonds are being used either to another carbon or to a hydrogen. Means that that molecule is probably not going to be polar, okay? Unless it has a different side chain, which we'll talk about, or functional group, okay? So again, these molecules are not polar. They do not like to mix with water. Think of oil and water, okay? That's the best way I can put that. So 3 representations of methane, a simple organic compound. So here we have methane. Methane is just carbon bound with 4 hydrogens, all right? And methane is what we use to burn, right, in the stove. This combustion or this use of methane again in the stove. So you have CH4 plus, the two oxygens and flame or heat will ignite, and then you have carbon dioxide and two water groups, okay? So one thing to note about methane, it's a very, very potent greenhouse gas, much more potent than carbon dioxide. Burning of methane is actually encouraged rather than leaving methane as methane. Again, these hydrocarbon bonds, what we talked about before, these bonds here can actually hold a lot of energy, right? So they can retain or stop or absorb the heat from the sun much better than the carbon dioxide bonds do, okay, right? So we talk about, we hear a lot about CO2 on the news being a very, very strong greenhouse gas. Methane is much, much more strong, okay? Much worse for the environment than CO2, right? So we encourage the burning to break them down in the water, and CO2, much easier on the environment. Again, methane is used in your stove if you have a gas stove. All right. So hydrocarbons is fuel. Energy-rich organic compounds and gasoline provide fuel for engines, and energy-rich molecules and fats provide fuel for cells. Okay. So as you can see, octane and dietary. So this is a triglyceride. So dietary fat look very similar, right? They're saturated hydrocarbons. Okay. And typically what we think of that, saturated hydrocarbons mean energy, lots and lots of energy. Okay. So if my carbons are full, right, we have nothing but a chain of carbons that have a lot of hydrogens attached to them. We know that there is a lot of energy there. All right. And we'll talk about sugars and fats in a little bit, but do know that those hydrocarbon bonds, these bonds here, so carbon and hydrogen, this is energy. Okay. A carbon bonded to something else means lack thereof or less energy, right? So carbon and hydrogen, much more energy here, right? So think of fats. Fats have twice as much energy as sugars by weight. And again, that is because their carbons are completely saturated with hydrogen. All right. So a functional group. So a group of atoms that form the chemically reactive part of an organic molecule. A particular functional group usually behaves similarly in different chemical reactions, okay? So here we have a hydroxyl, carbonyl, carboxyl, so carboxylic acid, an amino group, which is important in proteins, and then you have phosphates, which are important in DNA and ATP, okay? So we talked about the hydroxyl group, we talked about sugars having a hydroxyl group. So when we look at a sugar, you'll see that their hydrocarbons are not completely saturated. You'll have offsetting hydrogen and hydroxyl groups or OH groups, okay? But again, this gives different characteristics and different reactivities to your molecules, right? And either they can bind or join up or link with other molecules, okay? So for the phosphate group, the phosphates typically make that backbone of the DNA, which we'll see in a bit. The amine group, the amino group will be binding to another carboxylic acid group to link proteins together, right? So nitrogen will bind to carboxylic acid, the carboxyl group, to link proteins together. And then the carbonyl group in lipids or waxes, okay? You'll see this often in waxes or lipids, okay? So kind of just keep that in mind. These are the different functional groups associated with the molecules we're going to be learning about. All right. All right, so giant molecules from smaller building blocks. So a monomer, a chemical subunit that serves as a building block of a polymer, okay? So a monomer is a single subunit, right? So for example, glucose. Glucose is a single sugar subunit that makes up a much larger polymer, right? If you put a bunch of glucose molecules together, you will have either cellulose, starch, glycogen, right? Those are all going to be polymers, okay? So a polymer is a large molecule consisting of many identical or similar molecular units called monomers, covalently joined together in a chain, all right? And so you'll see the starch, you'll see cellulose, you'll see glycogen. They're forming this big chain network of these multiple monomers that are kind of linked together, okay? So a macromolecule, is a giant molecule formed by joining smaller molecules. Example, macromolecules include proteins, polysaccharides, and nucleic acids. And again, we'll get into these different molecules coming up. Okay, so a dehydration reaction. Okay, so I'll talk about this train right now. Dehydration reaction is a chemical process in which a polymer forms when monomers are linked by the removal of water molecules. Okay, so one molecule of water is removed for each pair of monomers linked. The atoms in the water molecule are provided by the two monomers involved in the reaction. A dehydration reaction is essentially the reverse of a hydrolysis reaction. Okay, so the dehydration reaction is going to be linking individual monomers together, right? Just like this train is linked together. Each of these cars is a monomer, we'll pretend. And them being linked together will make that polymer, okay? So think of each of these individual cars as a single glucose molecule, and this whole train is going to be starch, right? Because starch is going to be repeating subunits of glucose all attached in a linear molecule, okay? And that's all starch is. It's just a bunch of glucose, right? But there are different properties of starch when compared to single monomers of glucose put together, right? Glucose tastes very sweet. Starch tastes starchy, okay? But you can eventually break down this molecule for the individual glucose molecules, which, again, why starch has so many calories to it. Think of rice, think of potatoes, right? So again, glucose molecule, individual cars, and then we have them linked together, making that polysaccharide or polymer starch, okay? All right, so synthesis and breakdown of polymers. For simplicity, the only atoms shown in these diagrams are hydrogens and hydroxyl groups in strategic locations, okay? So here we have multiple chains, this large chain of sugars. This is a dehydration synthesis reaction, right? So when you have the hydroxyl group and the hydrogen that are joined together to make this polymer or this longer polymer, you have the removal of water, right? So think of OH and hydrogen coming together and they make water, right? H2O. The removal of this water, again, meaning the dehydration, so removal of water, dehydrate, synthesis, so synthesis is putting together reaction, okay? So dehydration synthesis reaction puts monomers or sugars or polymers together, okay? So monomers into polymers, again, through dehydration synthesis reaction. And this is true for all of these biological molecules that we're going to be talking about, right? In order to link them together, the monomers come together with the polymers, we have a dehydration synthesis reaction, okay? Dehydration synthesis reaction. And I'm going to repeat that a lot, but you do need to know this, okay? So the removal of water and the addition of the polymer, right? Or the addition of the monomer, rather. So removing the water makes the bond, makes the link happen, and now you have a longer polymer, right? Building a polymer chain, a polymer grows in length when an incoming monomer and the monomer at the end of the polymer each contribute atoms to form a water molecule, right? The hydroxyl and the hydrogen group. The monomers replace the lost covalent bonds with a bond between each other, okay? So the removal of the water allows for the addition of this single monomer, okay? All right, so building a polymer chain, a polymer grows in length when an incoming monomer and the monomer end of the polymer each contribute. Oh, sorry, that was from the previous one. Oh yeah, so this is the same thing. Okay, so there's also breaking of a polymer, and this is true with hydrolysis. So breaking a polymer chain, hydrolysis reverses a process by adding a water molecule, which breaks the bond between two monomers, creating two smaller molecules from a bigger one, right? So think of this as breaking down the starch, breaking down the glycogen, right? You add water to clip or break down or rip off those individual monomers or break down the polymer or the polysaccharide or whatever that molecule is, okay? So the addition of water is required to remove off individual glucoses from starch, okay? Right, the water is added, you add the hydroxyl group and the hydrogen group, again, breaking that covalent bond between the sugars, between the proteins, between the fats, whatever it is, okay? And this is true, or this gives the reason why individuals typically drink water when they're consuming food, right? We typically have that urge, that biological urge to drink water, again, because we require water to break down these polysaccharides, right? To break down these individual molecules, okay? We require water in order for our catabolic, remember our catabolic functions are going to be the breakdown of these molecules, right? For energy, okay? So we do need water to remove this. We do need water to break down our food, all right? All right, so hydrolysis or hydrolysis, a chemical process in which macromolecules are broken down by the chemical addition of water molecules to the bonds linking their monomers, an essential part of digestion. And again, referencing digestion, right? We do typically have the urge to drink something while we eat. Again, this is going to be that biological urge in order to add the water to the system to further break down these polymers into monomers. Well, here it's showing a dimer, which is just two monomers linked together, right? Yeah, so keep that in mind. All right, so our first organic molecule we are talking about today is carbohydrates or carbs. These are sugars, monosaccharides, the smallest kind of sugar molecule, a single subunit of sugar, also known as a simple sugar, right? This is like glucose, this is maltose. Those are all individual single sugars. Yeah, important to note. So single sugar. Isomer, one of two or more molecules with the same molecular formula, but different structures and thus different properties. Okay, and we'll see that right now in this diagram. So glucose and fructose are isomers, right? Both of these molecules have the same chemical formula, C6H12O6. So glucose is the exact same chemically, has the exact same elements, has the exact same carbon, hydrogen, and oxygen concentrations, right, in each molecule, right? So an isomer is referring to the same formula but different arrangement. So you can see here that the hydroxyl and this oxygen group is moved, right? It's flipped. So in glucose, you have the hydroxyl group shifted down, and here you have the carbonyl group up at the top, right? So you hear that carbon double bonded to an oxygen at the very top, and then you have the offsetting hydroxyl and hydrogen groups here. Fructose, right, has the functional group shifted, right? You have that carbon double bonded to an oxygen right in the center, and then you have that hydroxyl group moved above this carbonyl group right here, okay? So keep that in mind. So isomers, right? Glucose and fructose are isomers. Again, they have different properties as well. One of these sugars, fructose, is typically considered a sweeter sugar than glucose, right? Also, fructose in our systems needs to be converted into glucose, okay? We use glucose as our main source of energy in glycolysis, which we'll talk about later, all right? So ring structure of glucose. So you have a linear structure and a ring structure, right? And typically we'll have the sugars labeled as such, right? So you have the carbons all lined up with the offsetting hydroxyl groups, but it's not a proper representation of the sugar, okay? Sugars are typically going to be circular or in the ring stage, especially in water, right? So sugar is only shaped linearly like this when it's not associated with water, okay? And that's important to know, right? So in aqueous solution, sugars are in this form here, the ring form, all right? That's an aqueous solution, right? If you remember what aqueous solution means, it means in water. So it's a key to understand that property or that principle. right? And by the way, the majority of the sugar that you will see, or the majority of the sugar that we deal with, is in an aqueous solution. So many of the times that we actually um, have sugar in our bodies, or we are consuming sugar in a drink or in a food source, this is going to be in the ring stage, right? This is rare. This will be found if the sugar is dry or it's in a pack, right? That we're going to put into our coffee or something in the morning, um, right? So this is not typical. We find mainly the ring stage in biological systems, right? Because the majority of biological systems, or all the biological systems, utilize water. Okay, so this is an aqueous solution. Okay, so this is an abbreviated ring structure. So in the book, they'll show it to you like this, right? So this oxygen is referring to this oxygen here, which you'll see, right? So what typically you have is that one carbon, this carbon here, is going to be bound to that fifth carbon here, and that sixth carbon is going to be hanging out with that hydroxyl group hanging. So you'll see this with the deoxyribose or ribose sugar in DNA, or ribose in RNA. So deoxyribose in DNA and ribose in RNA. But you'll see that the ribose sugar will form like this as well. Okay. But again, ribose is that 5 carbon sugar, so a little bit different. All right. Oops. So disaccharides, disaccharide, or a double sugar formation, to form a disaccharide, two simple sugars are joined by dehydration reaction. In this case, forming a bond between monomers of glucose and galactose to make the disaccharide lactose. So if you didn't know this, lactose is actually glucose linked together covalently to galactose, right? And yeah, so lactose is found in milk products, especially mammalian milk products. There's a lot of people that are lactose intolerant or have the inability to break down lactose. So I believe the allergy to milk is or the allergy to lactose is very, very rare, from my understanding of that. What I have heard is that the allergy, or the inability to break down lactose, is that lactose intolerance that is referred to, right? So I think most people will say, I'm allergic to lactose. That means they can't eat it, or else they have terrible bouts of diarrhea and gas and indigestion, right? So people will say, oh, I'm allergic to lactose. No, many a time it's they have the inability to break down glucose and galactose, or they have the inability to break down or separate this disaccharide lactose into glucose and galactose, right? So going into that, being lactose intolerant means that when you consume this sugar, it is not processed in the small intestine or that duodenum as it goes through the digestive system, right? So when you don't process the sugar, this is a high energy molecule. You do have a lot of bacteria in your colon. And if you let the sugar go unprocessed into your bowel, you have E. coli bacteria in there that will love to eat this, right? They just will tear this up. If the lactose makes it to your bowel and feeds your E. coli go crazy. They start dividing. They start breaking the sugar down, overgrowth, and that's what causes a bloating and the diarrhea. Okay, E. coli grows extremely fast for a microorganism, even compared to other microorganisms. And once the lactose hits that area, that bacterium just goes gangbusters and just loves it. Okay, so it'll overgrow, right? So this is what we refer to as a lactose intolerance. It's not so much an allergy, where I hear a lot of students will say, oh, yeah, I am allergic to lactose. Many times it's not an allergy. It's more so you lack the enzyme that can break this bond and allow for glucose and galactose to be separated and then further processed by your body, right? This lactose molecule can then be present in your bowel, which allows for the overgrowth with your microflora in your gut, right? E. coli loves this. Boom, overgrowth, indigestion, and diarrhea. Okay. So just a heads up for that. So disaccharides. Most common sugar is sucrose, so table sugar, which is glucose linked to a fructose molecule. Sucrose is isolated from plants, sugarcane or beets. So the majority of the sugar, sucrose that we get, table sugar we get, is actually from beets, not from sugarcane. So high fructose corn syrup, so it's approximately 55% fructose. Fructose, again, is much sweeter than glucose, okay? Fructose is that plant sugar that we typically think of. And again, it just tastes much sweeter, hence why high fructose corn syrup is just, you put it in anything, it sweetens it up right away. It's like comparing table sugar to honey. Honey is way more sweet, okay? High fructose corn syrup, way sweet, okay? And you can taste the difference, right? So different sugars, again, have different tastes, different sweet, I guess, sweet thresholds, right? One of them will taste sweeter than the other. But yeah. So the revised food label displaying the amount of added sugars. So I believe the FDA, USDA, all the governing agencies to be have limited the amount of sugar added into manufactured goods, right? So it should count for less than 10% of the total daily calories in your on your food label. Okay, so added sugars have been kind of scaled back given the obesity crisis in the United States. So the FDA has implemented that 10% rule, right? It should not be added sugar should not make up more than 10% of the total calories. Okay. All right. So polysaccharides. A carbohydrate polymer consisting of many monosaccharides, simple sugars linked by covalent bonds. Starch, a storage polysaccharide found in the roots of plants and certain other cells. It's a polymer of glucose. Okay. We have glycogen. So this is a polysaccharide made up of many glucose monomers. Serves as a temporary energy storage in liver and muscle cells. And then we have cellulose, a large polysaccharide composed of many glucose monomers linked into cable-like fibrils that provide structural support in plant cell walls because cellulose cannot be digested by animals. It acts as a fiber in our diet. All right. So again, these are the big three polysaccharides we'll talk about throughout the semester. Starch, again, plant storage, right? And we've seen starch before in that previous lab where we've added iodine, and starch and iodine change that color. Again, this makes sense. We talked about paper money and how if you have a counterfeit bill, we use an iodine pin to check for starch concentration because, again, our non-counterfeit money, or the money that we have circulating, does not have starch in it, okay? Paper does because it comes from plants, right? And so plants will save their sugar molecules as starch, right? They'll link their glucose together to form starch. Therefore, this is why we get starch in a lot of the plant components, right? Including paper, right? Think of potatoes. Where do potatoes come? They're technically associated with the root system, right? Potatoes are buried underground. Starchy, okay? Glycogen is the starch for humans, okay? So we carry glycogen in our liver and our muscle cells, and again, it's for quick, rapid recovery of our glucose, right? We can store glycogen easily, much easier than having dissolved glucose floating around in our blood, right? You don't want that either. If you have free glucose kind of everywhere systemically throughout the blood, it's diabetes, right? You need to be able to pull in those sugars, store it, turn it into a fat, or keep it into retain it into tissues, right? And the way we retain that glucose in tissues is glycogen, right? Again, muscles and liver cells will be housing or holding the glycogen molecules. Again, for quick and easy access to the glucose molecules. Cellulose is a little bit different. Cellulose is mainly going to be, So cellulose is not going to be associated with energy recovery. Cellulose is cable-like fibrils, right? This cellulose is essentially glucose that is arranged in a way that's very, 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 very sturdy. Okay. Um, so again, starch and glycogen are easily accessed, right? Your body can easily rip off the glucose from these large polymers and utilize that glucose for energy, right? So plants will utilize starch. Again, they'll rip the glucose molecules off the starch and utilize the glucose as energy, right? Glycogen is the same way in humans or mammals or animals in general, right? We have multiple chains of these of the polysaccharide. Again, the body will rip off the glucose molecules as needed, right? Cellulose, this is not the case. Cellulose is a structural molecule. Again, these three molecules are made up of all of glucose, but the way cellulose is arranged, we cannot digest it, right? So this is going to be the roughage or the fiber that we have in our diet, okay? When you eat leafy greens, your doctor recommends you consume more fiber, right? Fiber is going to be, well, I guess it's going to soften your stool, but fiber is going to be unable to be digested by the body, okay? Not even the microorganisms living in your gut can digest cellulose. It's just a very, 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 very strong linked or a polymer that's linked glucose molecules together, okay? And again, think of it as like the bulletproof armor of glucose, right? It's just really, really difficult to break down. You might say, well, animals eat grass, animals eat plants. There are animals that do have the ability to consume plant matter and digest it, right? Like we do not eat grass and we cannot harvest the glucose from the cellulose from grass, right? Because all plants are made up of cellulose. But certain mammals or certain animals do eat plants or do eat grass or do eat leaves, and they do get energy from those that plant matter, right? Well, in reality, it's not their digestive enzymes that are being used to break down that plant, okay? That cellulose, right? So cows or goats, for example, have something called a rumen, okay? They're rumenoid animals, right? They consume that grass, they consume that plant matter, but those rumen or those chambers in their digestive tract house microorganisms that are specially made to break down, or specially kept there to break down cellulose, right? So essentially, the microorganisms do all the work. They break down the cellulose. They have these giant chambers where they have a bunch of plant matter and a bunch of microorganisms that are essentially fermenting there and breaking down the cellulose. And then they can absorb the sugar molecules or the energy that way through their digestive system, right? So again, cellulose cannot be broken down by animals. You require microorganisms, bacteria, to break down the cellulose polymer. Okay. All right. So here we have starch. Again, starch in plants is going to be a single linear, large, long linear polysaccharide linked together. So each glucose molecule is individually linked together in one single chain. So glycogen in humans is a little bit different. One of the reasons is why plants don't move. They don't require energy really, really fast. They can take their time utilizing the glucose or ripping the glucose molecules off of this polysaccharide, right? Humans are much different, right? We need multiple chains of glucose molecules. So you can see here that glycogen has multiple branching chains of glucose, right? So glycogen, multiple chains, starch, a single linear chain in a row, and plants, right? So glycogen, we have multiple areas where we can start breaking off glucose in order to utilize in our muscles or our body in the liver, okay? And again, this is because humans move, we need energy right away. We cannot wait and individually rip off these glucose molecules like plants can wait, okay? So again, multiple chains here when compared to starch, all right? Starch, plants don't need energy that fast. They can have it linear, whereas glycogen, multiple branches of glucose molecules where you can essentially start ripping the glucose molecules at 3 different locations here, right? quicker, faster energy. Makes sense. We move. Plants don't move. All right. So cellulose is shown here. So very, very strong structure. So you see cellulose microfibrils in a plant cell wall. Again, so this is a structural component for plants. Again, we cannot break this down. So you have the linear, very well organized glucose molecules all linked together, as well as the hydrogen bonds from each chain. Okay, so it's important to note that this cannot be broken down by animals. You need bacterium or other microorganisms to break this down. Mammals don't have the ability to break this down. Even cows and sheep, right? The cows and sheep have special microorganisms in their rumen to break down this molecule. Again, and you can see why it would be very strong or sturdy, very well organized, as well as these hydrogen bonds linked between each of these glucose molecules. So very, very difficult to get to, whereas you see here starch and glycogen have a lot of surface area. They have a lot of area where you can get an enzyme in there to break off the glucose. Okay. All right, lipids. So organic compound consisting mainly of carbon and hydrogen atoms linked by non-polar covalent bonds and therefore mostly hydrophobic and insoluble in water. Lipids include fats, waxes, phospholipids, and steroids. So hydrophilic, meaning water-loving, these are polar or charged molecules, right? So hydrophilic, philic meaning loving, and hydro meaning water. So again, these are charged molecules. So sugar, it is charged. Proteins are charged, right? Sodium chloride, hydrophilic, they're charged. They can dissolve or miss in water. So hydrophobic, so water-fearing, again, are non-polar molecules. These are going to be lipids or fats. Okay. So here's demonstrating the hydrophobic oil separation and vinegar. So here we have the oil and vinegar, which is a water-based acid, essentially. So again, oil will float. They do not mix, right? So in salad dressings, we need to shake the bottle before, again, to mix that vinegar and the oil, especially like in Italian dressings, right? You shake it really vigorously to mix, and then eventually it'll separate out again, okay? Because oil is hydrophobic and water, again, hydrophilic. Water likes water. All right, so fat. Large lipid molecules made up from alcohol called glycerol and 3 fatty acid chains. This is a triglyceride, okay? Most fats function as energy storage molecules, right? Fats are kind of difficult to break down, actually are difficult to break down by your body for the fact that they are not able to mix in water. We utilize water for the majority of our metabolic pathways, right? Water is essential in hydrolysis or dehydration synthesis reactions, but hydrolysis in order to break down fats, right? It's going to be difficult because guess what? Fats float in water. Fats do not like water, so it's difficult to break down in the body, right? So it makes sense why we would use this as a long-term storage molecule, right? Fat is very difficult to break down once it's in this form, especially in our fat cells or adipose tissue, okay? So again, once you got fat, it's hard to get rid of. This is why it takes, some people a long time to lose weight. Again, using the fats, it requires a caloric deprivation in your diet. All right, you need to be calorically deprived in order to even access these fats in the body. And again, it's because these are made for long-term storage. Short-term storage, think of glycogen, right? Glycogen is just glucose linked together. Fats are long-term storage, okay? So not easily accessed by the body because of their hydrophobic tendencies, right? They do not mix in water, but your body is made up of water, so it's difficult to break down those fats, right? And that's why, again, it's easy to store that for long periods of time. All right, so triglyceride, a dietary fat that consists of a molecule like glycerol linked to 3 molecules of fatty acids, right? So here we have a triglyceride. Here's glycerol. Again, a triglyceride is referring to the 3 chains of hydrocarbons, right? And these are saturated. Okay. Um, yeah. So a dehydration synthesis reaction, again, will link those fats together. All right. So this is that structure of a triglyceride. All right. So it's important to note that we have something called saturated and unsaturated fats. So what does that mean? So saturated means that we have complete saturation of carbon molecules with hydrogen. So what are some of the traits of saturated fats? I know that we hear about saturated fats being bad for you. We hear about saturated fats not being good, especially because they're associated with butter and animal fat. So saturated fats are essentially just saturated, right? And typically at room temperature, they're going to be solids, okay? So think butter, think margarine, think, well, I guess margarine's not a good idea, not a good example, but lard, right? Uh, so lard is a good example. Lard and butter, again, saturated. This means that they are solid at room temp because of this reason, right? These straight line hydrocarbons allow for the fats to sit very close together, therefore not letting them become liquid, right? They can sit and be solid at room temperature like butter, like lard, okay? And these are much, well, they're considered higher energy molecules as well, because again, they have more hydrogen bonds than an unsaturated fat. So an unsaturated fat is going to be like oil, so fish oil. Think of the oils found in certain types of peanuts or nuts, fish also. Okay, so unsaturated fats have breaks or kinks in the tails or these triglyceride tails. So think of the triglycerides here. So again, this is an unsaturated hydrocarbon. Again, that double bond means that it's taking a bond away from another hydrogen, right? So thus limiting the amount of energy when compared to a saturated hydrocarbon here, okay? The more double bonds you have in this fatty acid, the more liquid like this fatty acid will be, or this triglyceride with the unsaturated links. Okay, so this is going to be more like oil. Again, more double bonds, the more viscous or the less viscous it will be. So the more liquid like it will be. Okay, whereas if you have it completely saturated, it will most likely be solid. All right, so saturated fats more solid, unsaturated fats more liquid. Okay, and the liquid state is caused by these double bonds that are going to be throughout those fatty acid chains. Okay, those double bonds again will provide space, right? So if you look here, if all of these had multiple double bonds, you'll have spacing between those triglycerides. That space contributes to the liquid state of that of this specific fatty acid or triglyceride. Okay, so multiple double bonds create space, makes it more liquid. Let's say if this is completely saturated triglyceride, you'll have these three chains of fatty acids. If they're completely saturated like this, it will not be viscous. It will not be liquid. Okay, it'll be solid at room temp. All right. So the total fat in the same serving size from 3 different sources is broken down into saturated fat, monosaturated fat, unsaturated fats with just 1 double bond, and polysaturated fat. So unsaturated fats with 2 or more double bonds, right? And you don't really need to know the polyunsaturated fat or the monounsaturated fat. Again, I just need you to know the difference between saturated and unsaturated, right? So saturated, again, is going to be more like butter. Unsaturated, think oils. Okay, and again, we typically associate the butter or the saturated fats as being worse for you, right? The animal fats, the lard, the butter, things like that. Whereas the like vegetable oil or the canola oil, right? These are going to be a little bit better for you, you know, as in terms of what you should and should not be eating, right? Avocado, things like that. Okay. Yeah. So here we have saturated fats, right? These are typically the unhealthy fats, but these are the good, like, tasting fats. So you have it in dairy products, beef, butter, cheese, right? These are all the things that we love to eat, but we probably shouldn't have too much of. Okay. Unsaturated fats found primarily in fish and plants, usually liquid at room temperature, right? We have the salmon here, Brussels sprouts, different nuts. You have trans fats. Again, these are going to be bad as well. So these are unhealthy fats in hydrogenated process. So foods, solids, and room temperature. So plant and fish oils are unhydrogenated, usually liquid at room temperature. These are good for you, right? You don't want trans fats. Let me see trans fat. Yeah, I already talking about trans fats right now. So hydrogenation is the artificial process of converting unsaturated fats into saturated fats by adding hydrogen. Okay, so this can be a problem, right? So if you take these fish oils and nut oils and add heat, pressure, and hydrogen, right? So this is going to be called hydrogenated fats. Okay, you're essentially adding hydrogens to the double bond, right? Oops. So if you're hydrogenating this fatty acid here, you're adding two hydrogens here, okay? You're adding hydrogens. And what that means is that you're hydrogenating it, right? You're adding hydrogen, which is not good, okay? You're making this good fat into a bad fat, okay? And this is common because it's cheap, right? This is much cheaper to produce than this. Animal fat is way more expensive by the pound than plant-based fats or plant-based lipids, okay, oils, right? So much more time-consuming, much more expensive to make. Whereas if you can grab some canola oil, which is pretty cheap, and hydrogenate it and turn it to something similar to butter, you can sell that and people can flavor it like butter and put it in their cookies, right? Margarine, essentially. But we do know now that the hydrogenation process can cause issues, right? The body doesn't process the hydrogenated fats in a way that is good, right? So think of HDLs or high density lipoproteins and LDLs, low density lipoproteins. That's what when you go into the doctor and you get your blood work done, they take your blood work and your HDLs or your high density lipoprotein should be high and your LDLs should be low, okay? But what they found is that these hydrogenated fats, such as the ones you find in fryers, right? That makes sense because fryers or the ones you find in baking products, what they've noted is that actually can change your biochemistry, right? It can change what good high density or it can increase your low density lipoproteins and decrease your HDLs or your high density lipoproteins, which is not good, right? You don't want to flip flop your cholesterols, okay? But again, the hydrogenation of these oils into a solid state is not good for you, okay? So trans fats, an unsaturated fatty acid produced by the partial hydrogenation of vegetable oils, most margarines, many commercial baked foods, and many fried foods, right? So again, trans fats we know are bad. And trans fats are referring to how those hydrocarbons are added, okay? And again, trans fats provide an issue for humans during the breakdown process. This transformation instead of a cis formation of fat it also changes the biochemistry of our body, right? We don't process this well. Again, it's artificially made, and it's not good for our body. It's not natural, okay? Difficult for our body to process, all right? You don't find trans fats in nature, right? They're created in a manufacturing process. Therefore, it can throw off our biochemistry in our body, okay? So affecting our cholesterol readings and things like that. So steroids, they're a type of lipid with a carbon skeleton in the form of 4 fused rings, 3 6-sided rings, and 1 5-sided ring. Examples are cholesterol, testosterone, and estrogen. Right, many times we think of testosterone, we think of or we think of steroids, we think of just testosterone and estrogen, but cholesterol also belongs into that group, okay? So cholesterol is going to be a cell membrane component, and it's going to be making the cellular membrane rigid or sturdy, depending on what it's used for. Oh, excuse me. Oh, sorry about that. All right, so examples of steroids. The molecular structures are abbreviated by omitting the atoms that make up the rings. The subtle difference between testosterone and estrogen results in anatomical and physiological differences between male and female mammals, right? This example illustrates the importance of molecular structure to function, okay? So if you see here, this is testosterone and estrogen. They look very similar, right? And these groups here are these functional groups here are the things that are different, and again, they make very different characteristics, right? They provide the secondary sexual characteristics that you see during puberty. Uh, yeah, very similar structures. Okay, and we'll talk about cholesterol later when we get into cells. All right, so baseball player Mark McGwire is one of the many famous athletes who have admitted to steroid use. So steroids are kind of a big thing in sports as well as in human consumption, right? Many times we think of steroids in athletes, but steroids have been used by men for a very long time. I think from what I've read on the research with steroid use is men have been using hormones longer than women have in terms of like the birth control. I think the birth control was 60s, 50s, maybe. Men have been using steroids for muscle development and increased muscle mass for since the 30s and 40s, I believe. So so it's crazy why they haven't developed a male birth control yet, because this again, the testosterone can have negative side effects on sperm production, given that it'll allow the testicles to stop producing if you have disseminated testosterone in the body, right? And again, the book kind of talks about the negative side effects of steroids, the androgenic side effects of steroids. You don't really know this, but I'm just giving you some extra about steroids. But they talk about the negative side effects with steroids. I mean, the anger, the aggression, the male pattern baldness. You can have gynecomastia as well. So that is when you increase your male hormone, your body's natural response is to offset that with female hormones. So sometimes males will develop the breast tissue, given that the increased testosterone has also increased their estrogen, right? The androgenic side effects are going to be those side effects as well. Androgenic side effects are common with steroids or hormones, right? They provide the secondary sexual characteristics, right? The development of the musculature, the darkening of the genital region, the tissues in the genital region, pubic hair, things like that, right? But again, steroids are also they have a very bad rap in the news, but again, they're also very useful in treatment strategies, especially in burn victims, right? Or people that have suffered a very bad accident, such as a terrible car accident, right? Steroids can be used as by means to increase growth and tissue growth and cellular growth in the body, right? So like decaderoblin will be used in the hospital setting, right? Decaderoblin is actually a steroid that is very similar to testosterone, but it does not have the negative side effects of testosterone, okay? So decaderoblin can be used to mainly grow or promote growth in the body, but without having the male pattern baldness, the increased aggression, the gynecomastia, right? The breast tissue forming in men, right? So decaderoblin is a useful steroid in the hospital setting. By the way, if anyone's listening to this and they're like, oh, cool, well, decaderoblin is a steroid to take, don't take that either. For recreational use, start to get bigger. It will, provide you with larger muscles and growth without the negative side effects of testosterone, but it will also lower your natural T. So you will lose all your secondary characteristics, sexual characteristics, right? Your sex drive, you will not be as, you'll lose all your male, I guess, drive. I'll put it that way, right? So decadurabolin, great in the hospital setting for burn victims that need to grow and need to have the cellular growth and body healing, but bad if you're going to take it recreationally, right? So steroids, again, are great for individuals that need it, or individuals that are hurting or that are in the hospital that require some sort of extra help in recovery. Don't take it if you're just trying to get bigger muscles and perform better, right? You do have negative side effects. All right, so proteins, a biological polymer constructed from hundreds of thousands of amino acid monomers. Proteins perform many functions within living cells, including providing structure, transport, and acting as enzymes, okay? And enzymes we will talk about later in more detail. Some of the varied roles played by proteins, excuse me. So structural proteins provide support, so hair, ligaments, horns, right? So storage proteins provide amino acids for growth. So seeds and eggs are rich in storage, right? Albumin in eggs. Seeds have proteins, again, for storage. Contractile proteins, again, this is going to be your myosin and actin filaments in your muscles. Again, structural and contraction or movement in humans. Transport proteins. So proteins like hemoglobin with red blood cells transports oxygen, and then we have enzymes, right? So remember we talked about breaking down hydrogen peroxide, right? You pour hydrogen peroxide in your cut, and it breaks down the H2O2. Again, enzymes are important to protect against reactive oxygen species, as well as breaking down different chemicals, right? You produce amylase in your mouth that breaks down starch, right? So has anyone ever done the cracker in the mouth, and you hold it in your mouth and you swish it around and you let that cracker eventually break down? Because again, a cracker has a lot of starch. Eventually the amylase in your mouth, which is an enzyme, will break down the starch molecules into individual glucose molecules, again, making that cracker slowly turn into glucose and slowly get sweeter and sweeter the longer you hold it in your mouth. All right. The monomers of proteins, amino acids. All amino acids share common functional groups but vary in their side chains. So you have an amino group, you have a carboxylic acid group here, and then you have a side chain, okay? So again, the amino group will link to another carboxylic acid group or carboxyl group, okay? So nitrogen to carbon, or nitrogen to this hydroxyl group will be linked, okay? These side chains are going to give rise to the different amino acid groups, right? So this side chain for leucine, this is a side chain for serine. Again, you have hydrophobic or hydrophilic side chains. You can also have a neutral side chain. Again, the amino group will link to the carbonyl group, okay, or the carboxylic acid group, okay? So nitrogen, again, will bind to another carboxylic acid group, this OH group here, and they'll link together in long chains, right? So here you have a dehydration synthesis reaction, right? So the removal of water will create a covalent peptide bond. And again, a peptide bond is going to be between the carboxylic acid group and the amino group. You do need to know this, all right? So again, carboxylic acid group to the amino group linked together. This is a peptide bond, again, made through a dehydration synthesis reaction. Water is lost, bond is formed covalently. All right. So there are different structures to proteins, which we'll get into later, but we'll get into a little bit now, but mainly you'll learn about the different structural components later, and the primary, secondary, and tertiary structures of proteins. But again, for now, the protein, again, is going to be made of these peptide bonds that link all of these amino acids together in a long chain, right? So this is a primary form of that protein, okay? There is a secondary form, which is going to be the secondary folding of this protein. So it's going to be the alpha helices or the beta pleated sheaths. These are going to be the beta pleated sheaths here, but it's just the way the amino acids is going to fold, okay? So they'll fold in a different arrangement, and this will give rise to the structure of the protein, right? Here you can see how the polypeptide folds into a compact shape, and this shape will be the reactive or the useful form of that protein. The individual strands, if they're not folded appropriately, you will not have a good protein, or your protein will not form properly, right? And it will not perform the task that is needed by that protein, okay? And here you have just another 3D model showing the details of the protein structure, right? So if you have misfolded proteins, or the protein is not folded appropriately, right? So let's say if the amino acid sequence of normal hemoglobin, you could have normal hemoglobin polypeptide, right? This gives rise to the normal red cell, the normal shape, okay? If you change the structure, or you change or distort the shape, you can have a varied hemoglobin effect, or you can have an issue with the hemoglobin, right? This structure, this sequence is important, again, because it provides a function for the body, it provides a function for that red cell, right? It retains that oxygen, or holds that oxygen, right? So normal hemoglobin red blood cells of humans are normally disc-shaped. Each cell contains millions of molecules of the protein hemoglobin, which transport oxygen from the lungs to the other organs of the body, right? So if you change this shape, if you destroy this, or if you denature the protein, which we'll talk about later, but denaturing essentially means you're changing either the structure or the shape, right? Then you could render this protein useless. It will not perform this task appropriately. So another thing to mention too is we denature proteins all the time when we cook, right? If you've ever cracked an egg and threw it on a pan and it was clear and then it turns white, that albumin protein is actually changing the structure, changing the shape. It's no longer useful. Well, it's useful to us because we can consume, but no longer useful in the sense of making that chicken, or helping that baby chick grow, right? We have totally destroyed those proteins. They are useless now for creating life, okay? And same thing here with this polypeptide or this hemoglobin, right? If you change the structure, it could render this molecule or this protein useless, okay? So here we have sickle cell. So sickle cell hemoglobin, a slight change in the amino acid sequence of hemoglobin causes sickle cell disease. The inherited substitution of one amino acid, valine, in place of the amino acid glutamic acid occurs at the 6th position of the polymer, which is crazy. So one single amino acid change here, the valine, changes the structure and causes sickle cell disease, right? So this is genetic disease. Again, I'll keep saying this, which we'll talk about later. But again, one change in the amino acid can change the entire structure, right? And rendering this protein almost useless, right? This sickling of the protein, or this changed hemoglobin, makes a sickle cell again in this red cell, or causes the cell to sickle. This means that this cell is much more likely to rupture or break in the body, and which can be very dangerous for individuals with sickle cell disease, right? We'll talk about that trait later, but I was going to talk about mosquitoes and malaria, but you don't need to worry about that right now. But one amino acid, it goes to show you that one amino acid can be very, very important in the structural integrity of this protein, right? Whereas normal protein will be useful and provide the cells a structure, a strong structure, and allow those red cells to transverse the body pretty easily. Whereas if you have this sickle cell hemoglobin, you have this curved shape, and then that patient is suffering, or that individual is suffering for the rest of his life from sickle cell disease. Again, from one single amino acid change. One single amino acid change can distort or change that protein enough to cause issues for that person. All right. So another thing about proteins, improperly folded proteins or denatured proteins can lead to brain diseases, right? So prions cause fatal weight loss diseases, including scrapie in sheep. This is also a prion disease is also mad cow disease. So I don't know if any of you have ever heard of mad cow disease, but it's the disease that kind of came about in the early 90s. in Great Britain. So this was an issue because they were feeding dead, or they were feeding cows other cows, which is, you don't ever want to do that. Okay. Again, for this reason, you can develop prion diseases, right? So here you see the prion proteins. Prion proteins are essentially going to change other normal proteins into the prion structure. So I believe it's alpha helices, or beta, I think it's beta pleated sheets into alpha helices. Again, these prions will then form clusters in the brain or in the cell, and then you have the wasting disease that you see with mad cow. Okay, again, proteins improperly folded that will then convert normal proteins into other prions. Again, this causes spongiform brain disease. Okay, so you'll see pockets in the brain of these proteins. And again, very similar to how Alzheimer's occurs, right? You have the accumulation of these plaques in the brain. But again, this is caused by a prion rather than the beta amyloid plaques associated with Alzheimer's. Okay, so again, prion disease, proteins, think Alzheimer's. All right, so nucleic acids, last one, then we're done. Nucleic acid, a polymer consisting of many nucleotide monomers, serves as a blueprint for proteins and through the actions of proteins for all cellular structures and activities. The two types of nucleic acids are DNA and RNA. Gene, the unit of inheritance in DNA or RNA, and some viruses consisting of a specific nucleotide sequence that programs the amino acid sequence of a polypeptide. Most of the genes of a eukaryote are located in its chromosome, chromosomal DNA. A few are carried by the DNA of the mitochondria and chloroplast. Okay, so again, this is going to retain the genetic information. Remember, we talked about the central dogma a little bit. So DNA to RNA and then to protein, right? RNA will go to protein. Okay, so here we have it. We have the central dogma of biology, right? So we have a gene, DNA, RNA to protein. Within a cell, a gene, a segment of DNA, provides the directions to build a molecule of RNA, which can then be translated into a protein. All right. So here's a nucleotide, and we have a DNA nucleotide monomer. It consists of three parts. We have a sugar, right? We have ribose here. Again, this is going to be in water, so it'll be that circular shape. We have a phosphate group. This connects to the next nucleotide here. So a phosphate group will bind here. So this phosphate will link to another sugar at that carbon here. Okay, and then you have a nitrogenous base, right? This is going to be thymine, but it can be thymine, adenine, guanine, or cytosine, which we'll talk about briefly later when we get into DNA. Okay. All right. So you have adenine to thymine, guanine to cytosine. So these hydrogen bonds will form between this structure. This backbone of the DNA, which we call the double helices, is going to be formed by that phosphate group here. So this phosphate, again, is going to be that backbone of that DNA molecule, right? And then you'll have the binding of adenine to thymine, guanine to cytosine, again, across from each other, right? So adenine will bind to thymine in a complementary, inverted complementary strand, okay? So cytosine, so guanine to cytosine, adenine to thymine. Again, you'll have that bond forming from the anti-parallel strand from the other side of that DNA, okay? So anti-parallel just means that the shape of the molecule is flipped or inverted, and then they hydrogen bond to each other. And again, we'll get into this in more depth later, but just know the structural component of a nucleotide, and that this is going to be making the structure. Multiple nucleotides are going to be making this double helix structure. Okay, but again, we'll talk about that more in depth later. But here it shows the base pairing. So this backbone again is going to be that phosphate group, and then you have the binding of C to G, A to T. So adenine to thymine, guanine to cytosine. Okay, and those are going to be the base pairs, right? And here between these base pairs we have hydrogen bonds. So the hydrogen bonds are going to be between these groups here, right? Okay. So RNA. So notice that RNA nucleotide differs from DNA nucleotide in two ways. The RNA sugar is ribose rather than deoxyribose, and the base is uracil instead of thymine. So instead of thymine, we have uracil, okay? But another thing that to note is that we do not have, or we have an extra hydroxyl group here, okay? So this hydroxyl group is actually makes this molecule unstable, right? Whereas DNA will have a deoxyribose or lack that hydroxyl group. This allows this molecule to stack on itself and allow it to have that double helix shape, okay? The twisting of the DNA in the double helix is very, very important for its strength, right? DNA is much, much, much more resistant to damage than RNA, right? RNA can easily break apart, whereas DNA is retained very well, okay? And that's important, right? We use DNA as our storage molecule in our body. It writes the whole code to everything in our body. Each one of your cells has their entire genome inside of it, right? Each one of your cells has all the information it needs to make another one of you, which is insane, right? So it makes sense why we would have that information stored in DNA, because it's very, very stable in this shape, right? This double helix shape. And it's very stable in that, and for the reason that it doesn't have this extra OH group here, and it allows the molecule to form that double helix structure, okay, or that twisting, right? If the OH group was present here, it's much more difficult to form this double helix structure. It's much more difficult to have it organized in this way, okay? Yeah, so RNA, much less stable. We do not use this as a genetic storage component. Some viruses do, such as the coronavirus and the influenza virus, will use RNA as a storage molecule. But again, the viruses will mutate very, very rapidly, okay? All right. Any questions? I know that was a lot of information. Any questions? No? No questions. All right. Well, please let me know. Here, I'll go ahead and kind of give you a little bit of the lab stuff. So the lab is going to be the exact same topics. Let me see. Let me stop sharing real quick. Okay, so here you have the different labs for today. It's due August 27th, so just make sure you do these labs. They're pretty simple. The questions, I made the questions less weighted, I guess. I know it was half and half last time, but I think some people had issues with the questions. There's going to be a quiz this week, so I'll be uploading that on. I should upload it on Thursday. I might upload it sooner so you get more time. But for this assignment, you're going to run through the labs. Let me know if you have any issues, and then you should have a few questions to answer, right? And essentially what these labs are going to be going through are identifying different biological molecules. And this is a typical lab that I would run for my students if we were in class. Most of this information I just covered. But here's where it gets more lab oriented, right? So we can test for carbohydrates. We use Benedict solution. So one of the tests that you'll be doing virtually is using Benedict solution. So real quick, blue solution, there's no sugar. Green or yellow, traces of sugar. Orange, moderate sugar. And then if you have a red, you have large amounts of sugar, okay? So you can measure the amounts of sugar using this Benedict solution. You must heat this, I believe for a couple minutes or so, maybe 7 minutes. I forget the procedure specifically, but you do heat the reagent once it's added to the solution. Again, the virtual lab will guide you through that. So carbohydrates, again, polysaccharides used in energy storage. can be detected using iodine, right? And this is going to be starch. So there is a starch lab. We've talked about starch before. We've used it in the virtual lab, right? When you add iodine to starch, it turns this really black color, okay? And so you'll be testing that again in the lab. Again, that one of those 2-point labs. They should be fairly quick, okay? Proteins, we can use a couple of different reagents for testing for protein. So you have a Biuret reagent. Again, you add to a solution, you heat the solution, and you look for the purple change, right? So proteins can be detected using Biuret solution. So copper sulfate plus sodium hydroxide, again, will change that color into a purple solution. It will turn or stay blue if there's no protein being added. And I believe this is the exact test that's in the virtual lab, okay? The heating. So lipids, there's an emulsification lab that I didn't add, but there is a much simpler lab that uses paper bags. So you're going to be rubbing the oil virtually on a paper bag, and you're looking for that oil gloss stain that you would see. Let's say if you got fast food, right? You go to a fast food restaurant, you order burgers or fries, and the oil seeps through the bag, and you see the bag kind of transparent. Essentially, that lab is kind of running you through that. Okay. So yeah, those are the labs again, and those are the reagents. I'll upload this PowerPoint for you as well, so you can have it as a reference. Again, yeah, the carbohydrates and the proteins and whatnot. You'll have the reagents there. And again, we'll talk about emulsification of fats probably a little bit later, especially when we get into digestive system. Digestive systems, we'll talk about the bile duct and bile salts and things like that. Okay. So again, I'll upload this for you to have. All right. Any questions? Any questions? So just make sure you're completing the lab. Again, I apologize for this morning. I thought my email sent. That is my fault. So I'll take full responsibility for that. For those of you who turned in the morning and didn't get to see the Zoom link. So, but yeah, I'll upload this recording once it's done. And then please complete that assignment by the 27th. And I guess I will see you Wednesday. Okay.